Our final speaker in the session is Dr. Nick Schork. Nick got his PhD in the Department of Epidemiology here and was co-chaired by Mike. He's held faculty positions at Case Western, Scripps, TGen, and the like. And he is currently at HonorHealth working on integrating genomics and AI for precision health and medicine. So Nick, thanks so much.
Thank you. Well, it's an absolute thrill to be here, I gotta say. I tried to think seriously about how to balance anecdote with kind of science and stuff, and so I erred on the side of sharing my anecdotes tonight for the simple reason that I think you're gonna need a couple of drinks to follow the logic behind my career. Just to give you a little bit of a preview, and many of you probably know this, my father was a colleague of Mike's for a couple of decades, and I met Mike 40 years ago when I was a graduate student at the University of Michigan in the Department of Philosophy. And I wasn't doing AI or logic, I was doing 19th century European philosophy. And I happened to be a computer programmer in a medical school, got more and more interested in genetics. Mike was the go-to person. I asked him questions, and he actually took me seriously. That led to, a change in program, and again, I'll go through this tonight if people are interested. But Mike absolutely opened some doors for me that hopefully will show up in some of the slides I'm gonna say. Okay, looks like one of my slides. Go back, okay. So I have a couple of things I'd like to chat about. Talk a little bit about scientific impediments to translation, taking something from the bench to the bedside. So as you probably got from my little preamble, I'm pretty restless, I think, intellectually. I find it very hard to find a home in any one particular discipline, so I tend to bounce around. And as Mike E. pointed out, I have made changes in my career to kind of go with the flow, kind of my interests. And I know that's outside the norm in a lot of academia, but it's kind of been my mantra for the last little while. And again, I'll just thank Mike and Pat Pizer, my co-mentor, for kind of putting up with me as I kind of bounced around from idea to idea. So I'll talk a little bit about what I perceive as scientific impediments. Talk about genomics as foundational. Like how can we actually dig things out of the genome that might lead to better drug products or, say, diagnostics. And then talk about an area of interest of mine for about the last 15 years in built-for-purpose clinical trials. Probably heard about N-of-1 trials or bucket or basket trials. I got more and more interested in these because as we started to probe the genetic basis of disease, we just realized how heterogeneous disease was. And it doesn't make sense to think a one-size-fits-all medicine is going to work. Okay, so scientific impediments. So most of you know about the translational kind of flow of information. This is actually a rubric used by clinical translation science awardee sites, where they talk about the flow of information from basic science to kind of implementation science. You got to discover something. That's usually in the lab, obviously. Turn it into something. If it's a drug, try to understand sort of its preclinical properties. Test it out in humans, make sure it's safe, non-toxic. And then go ahead and interrogate its use in the population at large, and of course, understand its behavior when it's actually approved. And that has absolute benefits for populations. For personalized treatments, you can think of having a single patient if you want to figure out how to treat them, say, in the optimal way. So you have to diagnose them, understand what's the root cause of their disease to maybe open up insights into what is wrong at the molecular physiologic level that might be amenable to modulation through, say, a drug or whatnot. Then you got to come up with an intervention for that patient. And now you probably heard about all the developments in antisense oligonucleotides and other highly personalized treatments where this kind of paradigm is becoming a reality. Then you got to go ahead and test it. And with a single person, you're going to have to do an N-of-1 trial. That means collecting more measurements on your patient rather than having a big denominator of patients. And then you warehouse your results. Something very similar could be put together to develop products and whatnot for personalized prevention, but I won't go through that in great detail. Well, suffice it to say that genomics can give us a lot of insight into this area here, and everyone in the room knows this. I'd say we're getting pretty good at building databases and whatnot to house information that might emerge from studies of whatever sort. There's better clinical trials designs too, not just N of 1, but I had mentioned bucket and basket trials. So we're getting pretty good there too. Where we start to struggle a little bit is in these middle areas, I would argue. So drug choice, trying to figure out what is optimal to give someone. This has not been worked out yet, in part because we don't have adequate drugs to benefit everyone. But here's where I think all the action is. We just don't know enough human biology to kind of put everything together. And the reason for that is it's very hard to study humans. You just can't go and do brain biopsies every 5 minutes on a patient to see what's happening up there. Right, so we've got to come up with better ways of understanding human biology, whether it's genomically informed or maybe more downstream. You know, how could we modulate phenotypes that are of relevance? So I've thought seriously about this for the last little while. I had a little editorial in Nature about 10 years ago arguing, look, we know drugs don't work in everyone, so we've got to kind of change things around. And if you think about it, there's all sorts of reasons why a drug wouldn't work on someone. The person you're giving the drug to might be undernourished. So you don't think the drug would work if they're actually suffering from malnutrition. They could be dealing with a very serious debilitating condition. They could be exposed to all sorts of radiation. I mean, all sorts of things that if they had would ultimately compromise the ability of the drug to do its trick, right? We also know that there's a genetic basis to drug response, too. People differ with respect to pharmacogenetic variation, whether that's pharmacokinetic or pharmacodynamic in orientation. They might have a monogenic insult that could compromise the effects of a drug. What's growing in interest now is kind of the polygenic background that might compromise drug response and whatnot. So again, a big impediment to overcoming this is really a lack of sophisticated studies exploiting clinically relevant human biological variation to kind of get at this, or so I would argue. Right, and I've had a couple of recent papers describing this. So what about genomics? So it's fundamental. Again, this was 35 so many years ago when I was in Mike's lab. I had my thesis. It was on methods in genetic epidemiology. Again, Pat and Mike were my co-advisors. As you might learn tonight, if I get to tell you a little bit about my past, this wasn't a time when there were bioinformatics departments or statistical genetics working groups, really. There was a couple of people like Mike leading the charge. And for the people interested, you had to kind of flock to them. But it wasn't obvious like how you would craft your thesis in this area. Would it be more data-driven? Would it be more theoretical, et cetera, et cetera? Not a whole lot of precedent in this area. So I ended up putting a couple papers together for my thesis. One was on two-trait locus linkage analysis. This was a challenge Mike gave me. He said, well, do you understand the concept of peeling or the Elston-Stewart algorithm? Kind of calculating probabilities on big pedigrees. And there are tricks that people had proposed, but this was a pretty obscure place to play in. I mean, there was a handful of people on the planet that could follow the logic behind this and actually implement this, and Mike was one. And he sat down and we talked about this and said, well, let's figure it out for 2 loci, not just 1. So he encouraged me to reach out to people. One was Robert Elston. He said, why don't you just give Robert a call and ask him about, the Elston-Stewart algorithm. I said, the young guy, didn't know anything. Said, okay, Mike, I'll do it. I did it. We had a great talk. Suffice it to say, I implemented two-trait locus linkage analysis into the Mendel program with Mike. And I remember coming into his office after I had done this. And I said, Mike, I think I figured it out and it actually works. And he was like, really? Yeah. I said, yes, I think we got it to work. So ended up having a joint publication with Jurgat and Joe Terwilliger, who many of you in the room may recall. So all sorts of papers emerged from my thesis work. I won't go through them. This is kind of through the years, but a lot. One in particular, I had a serious collaboration with Eric Lander. He actually let me in the room, I think because he knew I came from a discipline background. Again, I was a pretty strange student at the time, kind of restless, like I said, but he let me in the room and we ended up getting along great and published a paper on how to dissect genetic traits using linkage. It was more of a review paper, but got a little bit of attention. Over the years, we've done some other things here. I'll give a shout out to a second generation person. I know you know this thing with Kevin Bacon, how many people removed are you from Mike? So Danny Fallon, who actually works with Mike Epstein right here down at Emory, was my student, and we worked on haplotyping and kind of relating haplotypes to phenotypes. And this is Danny's work. She went on and done great things. Like I said, she's the dean at Emory. But we actually were able to patent these things. And what's interesting about this, and again, I'll give a shout out to Danny because she was so diligent in this, is this was well before the GWAS craze. So this was about 1998 when we started this stuff. So we were thinking seriously about how to relate kind of haplotype variation to traits using linkage disequilibrium and stuff, and it, again, grew right out of my graduate studies. The other thing we did, not unlike Dr. Lin, was focus on annotating genetic variants. So we came up with techniques to try to predict how a nucleotide substitution might affect a transcription factor binding site or whatever. This is with former student Ali Torkamani, a really brilliant student who now works with David Goldstein in a company down in San Diego. But we had such success, at least at the time, and this is so naive by today's standards, right? We used machine learning models, state of the field at the time, to kind of predict these things, but we could basically kind of infer functional effects of genetic variants. So what we did is actually start a company at the time. This was with another U of M graduate, a former faculty member, a guy, Eric Topol, that some of you know, who was down in San Diego, worked with him for a long time. We put a company together and then ultimately sold it to Craig Venter, another person that I've had some interactions with. The one thing I'll just say, you know, I actually published papers with both Eric Lander and Craig Venter and got along with both. Craig unfortunately passed away just a couple of weeks ago, as you might know. But I think because of my training and other sorts of things, I got a foot in the door. People actually took me seriously. So I got to work with a couple of others in the community that were doing interesting things at the time. Okay, so what are we doing now? One thing we're focused on is generation of reference genomes. And there's a couple of different strategies, and I don't want to offend anyone's intelligence in the room, but just follow me for a minute if you don't know about reference genomes and their use in sequencing. So you typically have a reference genome, some representation of a genome, and you map reads to it from the sequencing that you're doing. Now, a problem with this is if you see deviations from the reference, either that's a sequencing error or it represents a bona fide variant possessed by the individual whose genome you're matching. So now you can create kind of pan-reference genomes where you can record the sites that reflect variant sites and better map your reads more reliably. So that's a pan-reference genome. The thing that we're most interested in now is more de novo assembly of genomes, where you may have a reference genome, you have your reads, but you don't use a reference genome. Instead, you kind of build up big contigs by looking at overlapping sequence. So this is computationally quite difficult, but I have someone in my lab, Guangfa Zhang, who's kind of an expert on this. So we put together a de novo assembly of a Native American genome. We also have a pan-reference genome. And what's really unfortunate about this is we used a sample in the Coriell repository, and we had buy-in from the tribal community in Phoenix. So we were building this out, had this, used this, verified as someone with Native American ancestry, did the de novo assembly, took 2 years in stealth mode because we were encouraged not to talk about this until things came out. Now, of course, it's done, and I'll get to the punchline in a minute. But we achieved a quality in our diploid assembly, de novo diploid assembly, that kind of rivaled the available publicly accessible reference genomes. So spent a lot of time doing this. Unfortunately, Coriell removed this sample from the repository, totally undermining our ability to get consent. So this reference genome is just sitting there collecting dust. It'll never get published. So that's unfortunate. And we used a tool that Guangfa developed. You can see we kind of compared it. We call it XSCAF. Here's XSCAF, kind of contig building. Here's what Hi-Fi-ism is doing, and the Salsa's other tools. So we put a few statistical tricks and computational tricks together to enable this de novo assembly. Another area I'm very interested in is kind of these polygenic risk scores that are big with everyone. Everyone kind of understands the concept. You just count up the number of variants that are associated with the trait in someone's genome, and that'll give a score that correlates with their predisposition to disease. A paper by Setka Theresen and group in Nature Genetics a little while ago showed the clinical utility of polygenic risk scores. You can capture people as susceptible to certain diseases as people that carry monogenic variants, say with BRCA mutations and whatnot. And there's been a lot of publications applying kind of polygenic risk scores. I actually was a reviewer of this paper, and they asked me to write an editorial. So interestingly, I recruited my father and son to be co-authors on this paper. So I don't know how many people saw this. This was some time ago, but there's my son, Andrew. He runs a lab in Denmark in neuropsychiatric genomics, done it for about 15 years. And of course, my father, Mike's colleague for many years, passed away a little while ago. But we had fun putting this together. I had someone email me, who will remain nameless, and say, hey, Nick, this was the cutest thing I've ever seen. You, your father, and the son on a genetics paper. And I said, well, geez, did you read what we said? We had all these. And he goes, I didn't say I was interested enough to read it. I just said to way up there, way down there. Okay. Well, so what we've done is thought of this concept and turned it on its head. Instead of looking at polygenic risk scores, we'll get data sets looking at longevity and create polygenic longevity scores. So we got GWAS on lifespan and created polygenic longevity scores, about 11 different ones. Applied it to the UK Biobank parental lifespan data. They all showed associations. This one kind of outlier up here was actually trained on the UK Biobank. We kind of did it as a control, but these others weren't. So we showed that these polygenic longevity scores were predictive of lifespan in independent databases and even things like parental lifespan. And we've also showed that the people who possess high polygenic longevity scores, so are predisposed to live a long life, are also avoiding certain diseases like Alzheimer's, CAD, heart attack, type 2 diabetes, but not all disease. For example, cancer isn't really associated with this. We also found, interestingly, that a higher polygenic longevity score resulted in less risk of dying from COVID. We also looked at kind of their utility, contextualizing polygenic risk scores. So for Alzheimer's disease, if you have the double whammy, the unfortunate double whammy, a high polygenic risk score for Alzheimer's and a low polygenic longevity score, then you're at the highest risk of developing disease over time. So we looked and found interactions there. We actually took polygenic longevity scores constructed, this is yet to be published, on super centenarians and centenarians. And of course, they sit where you'd like, above the mean in polygenic longevity scores. They're way up here, and they have less polygenic risk scores for Alzheimer's. We've also done what other people have done, looked at what genes those variants are implicated in, and the pathways, and find that they kind of make sense with at least respect to some of the associations like Alzheimer's disease and looking at neurogenesis and whatnot. Another thing we're interested in, not unlike Dr. Lin that we're working on, is trying to leverage annotations to do more powerful GWAS-seq-like studies. So if you can annotate a stretch of sequence, then you can relate those annotations to the phenotype beyond just the presence or absence of nucleotide substitutions, right? So how can one do this? Leverage alpha genome, again, as Dr. Lin had mentioned. Actually working with one of my former students, Ryan Toohey, who developed MPRAs, these massively parallel reporter assays where they can interrogate and look at the functional significance of like tens of thousands or hundreds of thousands of SNPs simultaneously. So building a database, trying to create tools that will take a stretch of sequence, annotate it, and then will relate the functional predictions to the phenotype, not just the presence or absence of nucleotides. So we're doing that. Another area that we're really interested in is trying to find sequence that's unique to humans. So Agnes Chan in my group and I, with Michael Hiller, collaborated with Evan Eichler and team in this paper. And this is one of these 20 zillion author papers. And our material was elaborated, I think, in Appendix 17 or whatever. So there were quite a few pages of supplementary material, but we got a few sentences in the main paper. But what we did was we did multiple sequence alignments with Michael Hiller's TOGA tool to find regions of the genome that looked like they were absent in species beyond humans, so stretches of sequence. And we found quite a few sequences that looked interesting. One was this FOXO3b sequence, which shows homology to the FOXO3a gene, which has been strongly associated in human longevity. Now, we're looking at the functional significance of this transcript, which is expressed with Rusty Grage and Kuo-Fen Lee down at the Salk. Okay, another area that we're interested in the genomic space is leveraging sequencing for pandemic preparedness. This kind of topic, as you know, is kind of shifting with the new administration. But we published this paper a little while ago where we thought instead of having an assay to interrogate each pathogen that someone might have, you just sequence everything from a nasal swab, right? So use kind of next-gen sequencing for that. And we showed that it works. We can pick up certain pathogens like SARS-CoV-2 in this, and there's many different applications. I won't bore you with the details, but suffice it to say that we tried to get funding to develop this in the United States, but for the reasons I said before, it just kind of fell on deaf ears. So we took the same proposal and submitted it to HEDIA or HERA, an organization in Europe, and they funded it within the first 3 weeks that they got it. So we got $25 million to develop this NGS-based agnostic diagnostic for infectious disease. Okay, moving right along here. I'm very interested, as I said, in many different things. And so one area that's allowed me to kind of pull this interest together is focusing on aging and longevity, because you can incorporate different diseases, different behaviors, all that. So I'm the director of something called the Longevity Consortium, where we have quite a few different projects. And the challenge is to try to triangulate the results of studies that are very disparate. So, we have GWAS results from epidemiology studies. And then we have kind of focused genetically engineered iPS cells with Rusty Gage and George Murphy. How do we pull those results together? It's not entirely obvious, but it's been a fun challenge. Okay, so I'm interested in trying to study drugs that can induce a healthy state, not just cure a particular disease or lower a particular phenotype. So we think the way to go about this is not to focus on a primary endpoint, which is the rule at the FDA, as many of you know. Instead, do multi-phenotype studies. So instead of the traditional clinical trial where you may have one phenotype, maybe depression or something, and then you find people that don't respond, and then you genotype them and then do another study, and Avogadro's number of years later, you might have a solution to things. We think maybe we could turn things on our head and study using these emerging technologies like wearables and dried blood spots and other sorts of things to get multiple phenotypes on individuals to see how their body is reacting to these drugs. The basic idea is the drug you consider as just a perturbation like any other in an experiment, right? All experiments, you need to perturb whatever it is you want. Well, the drugs perturb the human body. Let's see what it does to the human body. And we've talked about this a few times. Won't go into great detail about this, but well, we've got some stuff in place. So this is where these N-of-1 trials or SCEDs have had the opportunity to collaborate with many people in the field kind of thinking about these. There are many advantages to these. We can assess intervention effects if we aggregate these single case experimental designs or N-of-1 studies. We can assess variation in response. I'll give you one slide where we did a few studies exploring the power of aggregated N of 1s versus other kinds of, say, randomized clinical trials. You can assess causal relationships. You have kind of time series data. If you got the right data, you can use Granger regression and other sorts of tools to get at causality. You can validate surrogate endpoints with these sorts of studies. You can identify clinically relevant patterns when you aggregate them. And lastly, you can identify phenomena that benefit patients in real time. So this is instead of using the participants in your RCT as sort of guinea pigs to vet the drug, we're collecting data on the patients to see if the drug benefits them. And even if we find out the drug doesn't benefit them, if we've collected enough data on them, we might understand a little bit more about their disease. So we think we have a twofer, not only get some interesting biological insights, but benefit the patients at the same time. So here's the one slide I want to show. Let's say you could only afford to make 640 measurements on a phenotype. And you're interested in looking at the average effect of, say, an intervention on that phenotype. Should you get 320 individuals with 2 measurements, one before and after they get it? Or should you get, say, 10 individuals with 64 measurements, 32 before in kind of a time series fashion, and 32 after? Well, if they're independent and you're just interested in the average effect, then it doesn't really matter. But if you're interested in testing the hypothesis that there is variation in response, then it turns out the more powerful designs have more measures on smaller numbers of individuals. Now, this changes with the strength of the serial correlation. I won't go through that, but suffice it to say, it might be better to actually collect more information on patients in a longitudinally than getting a bigger denominator. All right. And we've looked at this in the context of multivariate phenotypes, sequential trials, test a patient, do the N of 1. If they respond, put it in a little bucket, test the next patient. If they're a responder, put it in the responder bucket, and just build up insight into whether the drug is actually achieving same frequency of response that you may have identified. So sequential aggregated trials. So what do some of these studies look like? Well, here's one. This was published a little while ago. Ali Torkamani sequenced the genome of a patient in one of our studies. This was also with Eric Topol when we were working together at Scripps. And this was a young child with a debilitating neurological condition, couldn't figure it out, diagnostic odyssey, sequenced the genome. Ali found the gene, ADCY5. We now knew what the gene was, but now we didn't know how to treat them on the basis of this. The patient was not only in a wheelchair, I'll show you pictures in a minute, but had very severe nighttime tremors, to the point where the flailing and the noise would wake up the parents. So they went in and started recording on their own the intensity and severity of the seizures that this young woman had, their daughter had. We went through the literature and found a drug, which is off-the-shelf drug that looked like it might abrogate this thing. Here's our N of 1 trial, don't need statistics. A lot of tremors up here, gave the drug, kind of disappeared. So didn't even do any real analysis on that one. We've since found that other things actually benefit the patients. This is Lily Grossman. She's been on record. She has a website about this. Now caffeine, high levels of caffeine actually have an effect on this condition. And you can see Lily pre-intense caffeine use and post-caffeine use. Here's another study, had a patient with a severe circadian rhythm imbalance, so could not sleep at night, and this caused tremendous disruptions. So what we did is ask what drugs she was on, a bunch of different drugs, and we weaned her off all the drugs and then put her back on in different combinations, the whole time measuring the quality of her sleep, okay? So here's just sleep using a simple wearable. So over about half a year or so, the different color coding, what combinations of drugs they're on. So the bottom line is, and this was interesting, she had the highest quality sleep when she was off all the interventions. So that was interesting. And we have all sorts of data. This was published there. Now, her depression was still there, but she slept better. So the real dilemma, what symptom do you want to focus on? Okay, a last kind of area is kind of these N of 1 or SCEDs in a dish. So this is in collaboration with the guy Stuart Lipton down at Scripps. Stuart's been a buddy of mine for a long time. With John Yates, where John Yates developed a single-cell proteomic assay where we could engineer the cells, introduce a mutation, in this case APP, mutations, and then asked the question, could we identify proteins that look like they mediate the influence of the exposure on an EFIS signature, kind of get at a cell phenotype relevant to the condition that we are interested in. So we did some analyses. This was published a little while ago. We could then make two hypotheses. The mutation changes the protein landscape, and that influences the EFIS, so kind of causal. Or it could be the mutation influences the EFIS signature or phenotype, and as a consequence of those changes, the protein landscape changed. So we could test these two hypotheses, and suffice it to say that it looks like the proteins actually mediated the mutation effect. And I won't go through all this, just suffice it to say we found some things that looked interesting that might be reasonable drug targets. Okay, last thing I'll say, a lot of interest in developing predictive models of one sort or another to decide who should get what drug, right? Everyone's doing this. Big initiatives like Truveta, 140 million patient records, they're doing this, blah, 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 blah. You can build these models, and they're all meant to kind of determine or at least guide what drug a patient should get. So we think, okay, well, we'll take some of these and actually put them to the clinical test. Like, we'll see if the predictions actually work. They recommend what drugs a patient might have. So we'll subject them to a single case experimental design of the type I said, see if the prediction actually worked. If it did or didn't, we'll feed that information back to the system and develop what amounts to a learning system, right? So this has been talked about, including people at University of Michigan, the kind of a journal devoted to this. So we think we can actually do this, and that's in fact why I moved to a healthcare system. We're actually trying to build that in the whole SCEDS and sequencing people with rare diseases in that. And in part, we moved there because Arizona State University is building a new medical school with HonorHealth as its partner. And the medical school is devoted to both medicine and medical engineering. So again, had all the bells and whistles, different devices, we can bring sequencing to play. There's going to be training the next generation person. This is Laura Getz. She's my significant other. She's a physician who is running the clinic, but there are certain advantages to where we move. So I will just stop there and thank you again. And thanks, Mike. And hopefully I'll give you these anecdotes tonight, which I think you'll enjoy. But